Hi everyone, my name's Elizabeth Hammond. I'm a third-year PhD student in the Department of Obstetrics and Gynecology. So my PhD project is looking at embryo biomarkers. So the reason why we want to look at this is to try and improve our IVF success rates. IVF, or in vitro fertilization, was developed as a treatment for infertility. And this may be surprising, but actually 1 in 6 couples will experience infertility. What's more, this is a rising trend as women delay childbearing to pursue their education and career. And we know that a woman's fertility falls as she ages, so that by the time she's in her mid to late 30s, her eggs are less able to make a baby. In vitro fertilization, or IVF, was developed as a treatment for infertility. And this little baby girl here, this is Louise Brown, and she is the world's first IVF baby born in 1978. So since this time, there have been over 5 million children born through the use of IVF technologies, and this number continues to rise, especially in the context of delayed childbearing. So what happens in IVF, or in vitro fertilization, is a woman is given drugs or hormones to promote her body to produce eggs. These eggs are then surgically collected and fertilized outside of the body with the partner's sperm. The resulting embryos then develop in a lab, and usually there are multiple embryos developing at the same time. These embryos are checked on once a day to see whether they look normal. Not all of them will make it, but by day 5 of development, just one of the embryos will be chosen to put back into the uterus, and the remaining embryos will be frozen for future use. So how do we choose the embryo to transfer into the uterus? So when we look down at the microscope at these embryos once a day, what are we looking for? So we know that the way an embryo looks is important. And when we check these embryos once a day, we look for whether they have a specific number of cells on certain days, and we look to see whether they are reaching their developmental milestones. The problem is that not all good-looking embryos, like this embryo here, actually go on to make a baby. And what this means is that more often than not, we actually choose the best-looking embryo to put back into the uterus, but this embryo that we choose actually never has the potential to go on and form a baby. And this means that our IVF success rates are still relatively low, with only 18% of our IVF cycles actually resulting in a live birth or a baby. What this means is we actually need better methods of choosing the embryos for transfer into the uterus, and this will essentially give patients their best chance of having a baby. So in this patient example, these 2 embryos here, or these 2 grey blobs, are from the same patient. So one of the embryos was chosen to put back into the uterus first, and this embryo sadly did not result in a pregnancy, causing a lot of grief and distress to the couple. The other embryo, on the other hand, was chosen second, and this embryo did result in a healthy pregnancy and a healthy baby. When you actually look at these 2 embryos, they pretty much look the same. And this means that there is something else going on that determines which embryo will make a baby, and this is something else that we can't see. And this is what my project is about. We aim to find improved ways of selecting the embryo for transfer into the uterus. And we're in search of any other clues that may tell us which embryo will make a baby, and clues more so than just looking at the embryo once a day. So in my PhD project, we have looked at this in 3 different ways, or looked at 3 different biomarkers of embryo health. Firstly, we have filmed the embryos as they develop. And with these videos, we're able to get a lot more information about these embryos compared to just looking at them once a day. With this information, we can calculate things like the timing of cell division. Secondly, we can look at the cells that surround the egg before fertilization. The health of these cells may translate into the health of the egg and then the health of the embryo. Thirdly, we can look at the little media droplet or the media bubble that the embryo lives in when it is developing in the lab. And we think that as the embryo develops, it may be releasing information into its surrounding environment or into this media droplet. So firstly, we wanted to see whether we could use a system called time-lapse photography to give us more information about which embryo will make a baby. So we put a cohort of human embryos in these time-lapse incubators that took pictures of the embryos as they developed every 5 minutes. So already we're getting a lot more information about our embryo development compared to just looking at the embryos once a day or once every 24 hours. So these images here are example pictures of what we get from this time-lapse photography. And you can see detailed images of embryo development. And with this information, we can calculate timing of cell division. So the time it takes the embryo to reach the 2-cell stage, or the 4-cell stage, or the 8-cell stage. What we found from this work was that our healthy embryos tended to show a strict developmental timeline. And when we saw variation or deviation from the strict timeline, it tended to mean that our embryos were unhealthy. What we concluded from this work was that our time-lapse photography may actually be clinically useful for the deselection of embryos for transfer into the uterus. So if we saw with this increased monitoring an embryo undergoing abnormal development, then we may choose to deselect that embryo for transfer into the uterus. Next, we wanted to see whether we could get information from the cells that surround the egg before fertilization. So these cells are called Cumulus cells, and they have a really important role in protecting and nurturing the egg as it develops. And we wanted to see whether the health of these Cumulus cells translated into the health of the egg and then the health of the resulting embryo. Specifically, we were interested in what kind of messages these cells could give us. And we analysed messages relating to cell health, cell development, and cell growth. What we found was that there were, in fact, correlations between the messages that we saw in Cumulus cells and then the future development or health of the embryo. However, when we tried to build a model that combined all of the information from the different Cumulus cell messages, we couldn't find any powerful predictors of embryo health, meaning that looking at these cells alone may not be able to tell us which embryo, or may not be able to give us enough information to tell us which embryo will make a baby. Next, we went in search to see if we could find any clues from the embryo media droplet. So this media droplet is what the embryo lives in when it is developing in the lab. And we wondered, as the embryo develops, could it be releasing information into its surrounding environment? And specifically, we wanted to search for genetic information or DNA. What we found was that there was, in fact, DNA being released by the embryo into this media droplet. And currently, we do not know the origins of this DNA or why the embryo would be releasing this into its surroundings as it develops. However, we are hopeful that it may be able to tell us about the health of the embryo. And work is currently underway to investigate this possibility. In conclusion, we know that looks alone cannot tell us which embryo will make a baby. And what this means is that we're often in a situation where we choose an embryo based on looks to put back into the uterus, but the embryo that we choose actually never has the potential to go on and form a baby. In our study, we have looked at different biomarkers of embryo health, namely time-lapse microscopy, Cumulus cells that surround the egg, and the media droplet that the embryo lives in. In our study, we found that correlations do exist between these biomarkers and embryo health, and our future work will focus on understanding these to work out whether they could give us more information to tell us which embryo will make a baby. And most importantly, better markers of embryo health will improve our IVF success rates, giving infertile couples their absolute best chance of having a baby and having a family. I would like to thank my supervisors, Lindsay Cree and Professor Andrew Schelling. I'd also like to thank members of the Cree and Schelling Lab, Fertility Associates, and also the Auckland Medical Research Foundation. Thank you very much for listening. Any questions?
I'm curious as to what happens to those droplets. If the embryo is releasing its DNA, do the droplets kind of go away at some stage, or are they do they end up being part of the baby?
Yeah, so that's a really good question. So the media droplets we're talking about, you can kind of think of it as the embryo is kind of in a swimming pool. It's quite a big droplet that we create for the embryo to live in while it's in the lab with us. What the embryo actually does right before we transfer it into the uterus is it does actually have this phase of growth and expansion, and what happens is the inside of the embryo actually forms this liquid cavity. So that liquid cavity, or that droplet of liquid, is part of the embryo and part of the baby. But what we're studying is the external environment that the embryo lives in when it is in the lab.
So you produce the droplet?
Yes, so it's a droplet. It's filled with nutrients that the embryo lives in while it is in the lab. And normally when the embryo gets transferred into the uterus, we would normally discard that media because we don't need it anymore. But this work could potentially mean that we could get some kind of useful information from that media droplet.
So you keep referring to 18% as though it's a fairly low, it's a low success rate, but I suppose the only reason people use IVF is because they have trouble with fertility. So I'm guessing like there are other issues with the mothers. Maybe the mother's unable to, is barren, or maybe there's issues with genetics. I had a question or concern. If there is genetic information available in the media, have you considered actually testing whether there's faulty genes? Or genes we could change?
Yeah, so that's a really good point that you raised. So we know that a lot of the reason why these embryos actually have such a low success rate in actually forming a baby is because they've got underlying genetic abnormalities. And this genetic abnormality is what actually underlies the decline in fertility as the female ages. So this becomes more and more pronounced as the female ages. So in terms of whether we could test this genetic information, that's exactly what we would want to do. So we would try and test whether that embryo actually has the normal genetics that we expect, the normal genetics that's compatible with life and therefore can go on and form a baby. So that's the idea behind trying to seek this genetic information with which to test and try find meaningful information about the embryo.
I'm just curious, in one of the slides you mentioned that for the time-elapse experiment, your healthy embryos stick stricter to their time of cell division, but how do you classify the embryos being healthy or not? Do you put it back into the uterus of the mother?
Yep, that's a really great question. So with the developmental timeline, we were actually really surprised because the embryos develop on such a strict timeline, suggesting that this is obviously really inbuilt to the program of the embryo, and the divisions that you see undergoing a strict timeline are compatible with life. In terms of in the literature how this has been tested, so the gold standard of testing this is relating the embryo health to pregnancy and then live birth, so whether the embryo forms a baby. In our study, it was a pilot study, so we looked at the health of the embryo right before it became transferred into the uterus. So as I said before, this is when the embryo starts to expand out and form a cavity. So we were testing these early timings to see whether they were actually predictive of later embryo development. And this is actually clinically relevant because you actually want to transfer the embryo into the uterus as soon as you can back to its natural environment. And also in some patients you might do the transfer of the embryo much earlier, so you'd only have a snapshot of its development and you'd want to try and predict what's going to happen to that embryo later on. So that was the whole idea.
You mentioned a second embryo that was transplanted into a patient and the baby, and it was successful. Had that second embryo been frozen? And if so, is freezing the embryos, does that degrade them in any way?
Yeah, that's a really great question. So that example of that patient, I used that example because both those embryos had been frozen. what happens most of the time is that your best embryo will get transferred in what we call, it will be fresh, so it won't be frozen. And then subsequent embryos become frozen. The freezing technology that we have nowadays, it's called quick freezing. We call it vitrification. So it's actually a very good technology nowadays, and pretty much 99% of our embryos survive this freezing process, which I think is pretty amazing. And in terms of the success rates, they're pretty similar between whether the embryo has been frozen or not been frozen. So it's more of a tool for us to transfer one embryo at a time and save the rest for subsequent cycles and subsequent transfers. So it's a really great tool.